Great speakers today, coming us from New York University in
Thank you very much, and it's a great honor to be here at this amazing campus and talk to you. And I apologize that we're all so late. The traffic in Dubai, you know how it gets. But thank you for waiting. So hopefully the color will get better. I'm going to take you on a little tour. I think we were invited to talk about our current research, but also how we got to it. So let's see. So what I'm going to talk about today is I'm going to explain why I have a picture of a fruit fly and an alarm clock, because that's what we're studying in my lab. It's the internal clocks for fruit flies. But I also want to tell you how I got to this point. And then at the end, I just want to give a tiny bit of advice to young researchers. Because I know that many of you are interested in thinking about research, and give you a few little tips that may or may not be useful. You have to decide for yourself. So, so I'll just keep talking. I'll start with, I want to kind of tell you some of the key moments that shaped shaped the direction of my career. And I think I was about 15 when I first came across this phrase, genetic engineering. And I had no idea what it was, but it sounded so cool. And so I did discover what it was, and I kept going. As an undergraduate, I was kind of torn between two ways, between kind of studying really basic science or somehow applying that to cure something. And so in particular, I was kind of a little bit torn between molecular biology or parasitology. Those were the two things that fascinated me. And in the end, I went towards the basic science route. And so I ended up doing my PhD in London, as Fatma just mentioned. And what I was studying was very basic questions of how genes were regulated in mammalian cells. And going into my PhD, I had this plan. I thought, over time, it would be a beautiful kind of smooth transition of collecting data, increasing data exponentially, and then I finished my PhD in 3 years, which is typically what happens in England. It didn't quite work out with that, as you might guess. So of course it took longer. So it took me 4.5 years. And the first couple of years was pretty much like this. It was a flat line. I tried quite a lot of different projects. And at some point I had this kind of we called it a midterm committee meeting. And one of the faculty suggested that I just get involved with one of the projects that was really exploding in the lab, and finally we managed to make a little blip and start getting some data. And from there, then it was very, very exciting. And then I kind of really hit the super, I forget what's the right term, but anyway, very rapidly accelerating, and finally kind of plateauing and getting my PhD in about 4 years. And this phase, this kind of, this discovery phase was so exciting. It was, I was literally doing experiments, finding out new things, testing, repeating it, finding out more things. That didn't quite come out. It looked good on my computer. But what I was doing, what I was meant to show you was I was using the existing data in the literature and putting that together with my research and really coming up with some new ideas, interesting ideas for how genes were regulated. And it was a very exciting time, and it seemed to kind of make these first 2 years of not much happening worthwhile. But at the end of that, I decided I needed to work on something a bit different. It was 1995 when I went to a transcription meeting at Cold Spring Harbor Labs in New York, and I could hear the detail with which everyone was learning about the mechanisms of gene expression, and it was just too detailed. And I decided to keep going in research, I wanted a new question, and I wanted something bigger, more open, where there was just less explored, and so I decided that the brain was actually quite interesting, and I was kind of torn between two, and I guess I also wanted to keep my kind of genetic or gene-based background too, so I was really interested in this idea of genes and how they could control behavior. And there were two fields I looked at. I looked at circadian rhythms and memory. I actually think in the long run memory is much more interesting, but the particular labs where I interviewed, I could see what I would do in a circadian rhythm lab, and the memory lab, it just wasn't quite right. So I ended up working on circadian rhythms, and that's what I've been doing since 1996, which is a long time to study something. So this is where we get to the idea of flies and alarm clocks. So I'm hoping that you know what circadian rhythms are. Do you have a sense of, do you know what a circadian rhythm is? Yes or no?
Yes.
It just, no, fine. Anyone that shakes their head, and I have to explain it. So let's see. So here, actually, this movie, I don't know that it's going to run. So it's not my computer. Oh, that's a shame. No, it's not going to run. If I get it off, I put it somewhere, didn't I? I just don't know how to use a PC. As a professor who doesn't know how to use a PC. Can you? But luckily everyone else does. So there was a movie that I uploaded. It was called Sunflower. So what we're going to watch is a movie over 24 hours of a sunflower. Yeah, you can just press play, thank you. So this is a sunflower filmed in real time in a backyard somewhere in America. And what you see is it's sunny and the leaves are up and the plant is busy photosynthesizing. And then as night falls, what you're going to see is that the plant actually starts, its leaves will actually droop. You can see that happening now. Because no one's moving this plant, really. And then you get to night. And now what is the circadian part of it is that the leaves start to come up again. They're anticipating when the sun is actually coming. And the idea is that they're kind of ready to maximally use all that light that's coming from the sun. So circadian rhythms is a rhythm of about 24 hours. We have them. You know them if you ever travel across time zones and your internal clock says get up at a certain time of day, even though it's the middle of the night. How do I get back? Oh, here we go. PowerPoint. It's not that hard, is it? So this is basically the internal clock of animals and plants. And I'm not going to talk any more about plants. I'm going to talk about fruit flies. And so this is what we do in my lab. How do we study a fruit fly's internal clock? How do we know what time it is for a fruit fly? What we do is we put flies in these small little cuvettes and basically have this infrared beam here, and every time the fly crosses the beam, that gets recorded by this detector down here. So we can record the activity of a fly for as long as we like. And in this particular experiment, showing you the activity of a fly recorded over about just a little bit over a week. And what you can see, what you can see is black is activity, and the higher that black bar is, the more, that means the higher activity is over that 15-minute collection window. So you can see that flies actually, and this is a 48-hour scale across here. So flies, they get a little bit active in the morning, they have some kind of siesta in the afternoon, they get very active around the evening, and then they have a bit of a sleep, and then they go on the next day. And if you were to get a computer to analyze it, it would tell you that this fly's internal clock is running at 24 hours. So this fly has a rhythm close to 24 hours. It's actually slightly shorter than 24 hours, but that's what the fly's clock is doing. And what the field of Drosophila research is based on genetics, finding things, to learn how something works, you often look at how something goes wrong. And so a long time ago, these mutants were isolated that no longer have beautiful 24-hour rhythms. This fly still has balance of activity and rest, but it no longer is organized into any coherent rhythm, and this is a rhythmic behavior. Whereas these flies obviously have rhythms, they're just not 24-hour rhythms. And what I forgot to tell you, which is really important, is that this is all happening in complete darkness. So this is the internal program, the genetics of the fly forming a clock, and that's telling the fly what length its day is going to be. So this fly has a day of about 19 hours, and this fly thinks a day is about 29 hours, meaning it takes 29 hours from waking up on this day to waking up on the next day. So back in 1996, we knew a couple of genes. So this gene that's mutated here is called period. In 1996, we knew a couple of genes, period and timeless. A couple of years later, there were 2 more genes, and we started to figure out how they work together to regulate the clock. And then we added a few more transcription factors, and then there are some kinases and all kinds of, there are basically probably about 10 or 12 proteins that form up this core clock. And this is all very nice, this is flies, but it turns out that we have very similar clocks working in our own brains to drive our own internal clocks. And in fact, in mammals, we have clocks in many of our tissues. So our liver will be going through circadian rhythms. And so we show different sensitivities to drugs at different times of day. And yes, we know these are important because there are actually a few people who have non-24-hour clocks. This is a woman from a large family, and she has a mutation in the human HER2 gene. And here in black, instead of looking at activity, here they're actually measuring sleep. And so this is her sleep, and you can see she looks a little bit like one of those short period fruit flies. In terms that she has a clock running at about 23 hours. So this, again, now we get to this point again where we're at some very detailed level. And so what I decided to work on starting my lab at NYU was to move into more of the kind of neurobiology of the clock. So how do we go from these transcriptional loops into actually behavior? And so a few of the things we're working on is looking at how these neurons work together. So what you can't see, unfortunately, because the blue channel is not working, is that each of these neurons is connected together and sending the process up here. And so the blue won't work at all. This is a disaster. Oh, yes, you can. All right, so then it turns out what we've discovered recently is that these neurons, which are called morning clock neurons, send a signal up at dawn to the evening clock neurons, and then the group phenomenon. They're working as a team. So that's one thing we're really interested in. Second thing we're really interested in is how actually, I can explain it here, how does the clock in these cells tell them to fire in the morning? And so the approach we've taken there is to identify those cells in the brain, and then we dissociate the brain into single cells so we can actually identify which of those clock neurons, and that's the one. That's one of them here. We can purify those on a fax machine and end up with a couple of hundred of those cells from which we can generate patterns of gene expression. And that's what I'm showing down here. And then the idea is to take these genes and ask which of these ones are rhythmically expressed and which might be controlling the firing of those neurons. And so can we make the link between patterns of gene expression and this rhythm and behavior? And we made an interesting finding, which is one of these genes, which is rhythmically expressed in these neurons, and it regulates the firing level of these neurons. When we change it, we alter the period as well. So we were expecting these neurons, these outputs, to go to be just a pure output to control the way these neurons fire, but it turns out it actually feeds back to regulate the core clock. So this has always, in science, has led to kind of another project. So now we've gone from how does the molecular clock regulate neural activity to a third project, how does neural activity feed back to regulate the molecular clock. And then one of the last pieces we're studying is actually, and again you can't see them, but again these neurons that project up to the top show rhythms in their structure. So every 24 hours they show a cycle of growth and retraction. And that's, now you can see this. So basically at dusk when they're not signaling, those neurons are kind of stuck together, and then they go into a much more expanded state at dawn with a high level of signaling. So we've been interested in how is that controlled at the molecular level. We've identified a few proteins involved in that, but also what's going on with the synapses, with the connections that they make, and why these neurons have to go through these quite big changes in structure every 24 hours. And that's something I can't really answer that because we're still working on it. So that gives you a taste of some of the projects going on in my lab. And I just want to finish with just a few tips. And maybe this is arrogant to think that I can do this, but hopefully this is useful. So, first of all, you have to try research in a lab. To know if you're going to like research, you've got to try it out. And that maybe you've got to try it in more than one lab. So here I've got someone with a microscope at the beach, we're actually not sure where this is, and someone in a more conventional lab. You've got to find out what you become passionate about. You may have to travel. I've traveled quite a lot with my research. It's been great, but I've met all kinds of different people. But to find a lab that where I was going to find the question and the people with whom I could work well, certainly being prepared to travel. I would say at the beginning, just be happy to be an apprentice, to learn what it's like to do a good experiment. And you don't necessarily have to have your own enormous questions at the beginning, but join part of a team. The science is really a team effort. So come in, work alongside someone in the lab, and hopefully you'll learn some of the elements of being a good scientist. But even while you're doing that, you've got to ask questions. You should always know why you're doing something. And so I think asking questions, that's what science is all about. How does this work? Why does it work this way? And so you can start that even as you're working as an apprentice, just kind of being, trying to understand why do we pick this particular technique. And then finally, and this is very important, be patient. That's absolutely key in science. And so that's where I'm going to start.
Thank you for your introduction. My name is Rafael. I've been here in the UAE for about 2 years now, and it's been quite a journey for me. I travel a lot actually I was born in South Korea, and I studied in Germany, and went back to Korea to work, and then went to US, and now I'm in the UAE. It's quite a journey for me, and I really happy to be here and to speak about my experience. And the title of my talk is, as you can see here, my journey from the macro to the micro and nano world. So I was working as a mechanical engineer. I got my training in Germany, and as you know, in Germany, the engineers get really traditional training as a mechanical engineer. I have to study all this, stress and load analysis of shafts, of different gearbox. So I did very traditional engineering as undergraduate, as graduate student. So I just worked in a factory. I would spend 6 months in a German factory to work as an apprentice actually. So I learned everything by hand, just doing building and on the lathe and the milling machine. I even did welding, and I learned all these traditional mechanical manufacturing techniques. And so I was working as a mechanical engineer and built this macro scale devices, tools, components. Then one day I saw this picture, and I said, wow, this is so cool. Especially the size of this gear. It's amazing. I said, wow. How can one build such a small, tiny gear? And I was really fascinated. I said, wow, I have to learn more about this technique. And I searched online. At that time, there was no Google, but I searched like in the library and online, and found out that there's a technique called microfabrication, which allows us to build tiny, micro-scale devices and objects. So I became more interested in this technique. I said, okay, this is something I would like to do. This is so exciting. So I just switched from macro world into the micro world. That's the transition I made. And I sort of looked at different research articles and found out that you can use this microfabrication technology to biological applications. And I thought, oh, this is something so cool actually. There's something like a really exciting research topic, and I want to study more about this. That's how I got started with research topic called BioMEMS. So MEMS stands for micro electromechanical systems. We just add bio in front of this term. So we apply this micro electromechanical systems to biological applications. And as I understand, you are from engineering background, but interested in applying these engineering skills in biology or in medicine. So this is really a perfect, I had a similar motivation when I started with BioMEMS research. Does anybody know what this is? Any idea? Any guess what this could be? This actually opens the world of micro.
It's the Leeuwenhoek microscope.
Yeah, this perfect. This is the first microscope actually, invented by this Dutch engineer or scientist, Leeuwenhoek, in the 17th century. With this very simple tool, I mean, you can see here, this texture here. and you have like a X, Y sort of stage where you put the specimen high. And using this simple tool, you could discover the micro world actually. You could see all these microorganisms nobody has seen before. So he opened up the world of microbiology. And this is how he used this tool actually. He just put this microscope against his eye, and you could really see all these tiny, tiny objects. So this was in the 17th century. In my research, I'm interested in applying this micro and nano technology to manipulate different objects. Cells, you can see the red cells, they're typically, around 10 microns in scale. So we use sort of micro scale devices. You can see here this structure in the background this is 1 micron in gap size, and this is about 2-3 microns in gap size. So we built this micro scale structures to manipulate. So what do I mean by manipulation? We want to separate cells by their sizes. There's population of cells, and if you are interested in isolating the specific cell population, and one of the markers could be the size, or it could be the flexibility. The red blood cells have a specific flexibility compared to other cells. You could sort of use this deformability of red blood cells to separate them out of the complex mixture. So we use these microscale structures to manipulate or isolate cells. You can also use these structures to separate or detect bacteria. Now, if you go look into lower, smaller scale objects, such as DNAs, proteins, you have to build nanoscale structures, which is shown here. So typical structure we're using to manipulate molecules, biomolecules, are between 10 and 100 nanometers. So we can build such a nanoscale object inside a clean room, and we can use this nanoscale tool to manipulate DNAs in different proteins. And if you build even smaller nanoscale tool between 1 and 10 nanometers, you can even manipulate ions. So you can control the transport of specific ion through this nanoscale channel. Now, how do we build such a nanoscale object, tool? Does anybody know what this is? So first, ENIAC computer, and this is the modern microprocessor from Intel. Do you know what enabled to shrink down the size of such a big computer to this scale?
Conductors?
Well, yeah, so you make this transistors.
Yeah.
But what is the enabling technology to shrink down the size of this huge computer to this?
Nanotechnology.
Nanotechnology. Can you be more specific? What kind of nanotechnology? Have you ever encountered such a technology?
Quantum physics.
Quantum physics?
Semiconductors.
Yeah, you're quite close. Semiconductors. How do you make the semiconductor tools, semiconductor chips?
The material you use.
Nothing etched.
You have silicon, right? You have to etch, right? That's the right keyword. Exactly. You have to go with the clean rooms. Has anybody been in a clean room? This is one of the modern clean rooms. People who wear this bunny suit and gowns and caps and so on. This is a tightly controlled environment with less particles than in normal environment. And you go into this clean room and use all these tools to fabricate such a microprocessor. So this is like one of the clean rooms you can probably find in Intel or in AMD or in any modern sort of factories. So we borrowed this technology to build our micro nanoscale object and tools to manipulate cells and so on. So we just borrowed the same technology, right, from semiconductor industry and apply this technology to biological applications. So what's our final goal? This is a traditional lab. You can see here we have different benches and you have different instruments, and you move one, after you do one step, you move this specimen or sample to the next station, and so on. So you move this, let's say you have a blood sample, and you have to cleaning step first, and then do the separation, and so on. And you just go through those steps, and at the end you do a detection and see whether there's any biomarker inside the blood. Our goal is actually to shrink down this size, the size of the traditional lab to this size, by borrowing the semiconductor technology. So we borrow this technology, and our goal is actually to shrink down this size as somebody from this side to this size. As you can see here, there are tiny channels here on this chip, and these tiny channels, we call it micro channels, they connect the one reservoir to the other one. So we can have multiple channels with multiple reservoirs connected, everything integrated on this small scale chip, and do all these processing steps on chip. So this will allow us to, A, reduce the time one needs to process a sample, right? Instead of moving the sample from one bench to the other one, we can move everything from one reservoir to the other reservoir quickly because the distance is small. B, the sample amount could be reduced drastically. Instead of using, let's say, a few milliliters of your sample, you can just use 1, maybe 10 microliters of your sample to process the same thing on this chip. And most importantly, this chip can be completely automated. So there's no more human intervention needed. You just put your sample into the chip and do all these processing steps on chip without any human intervention. This will allow us to reduce the human errors and reduce the time, detection, and so on. There are many benefits to do it. So this is one of the clean rooms we have built at NYU. So we have built a state-of-the-art clean room facility at our new campus. It consists of a spin coater here in the corner where you can deposit a thin photoresist on a wafer. And then we have a mask aligner to pattern the microstructure through a mask using UV light. And then we have a developer here in the chemical fume. And we have also a film evaporator to deposit a thin gold layer. And we have a reactive imager to etch gold or silicon structures. So using this clean room facility, we are in a position to build such micro nanoscale objects. Okay. So what I'm doing right now in my lab is I'm using a technique which allows us to concentrate the molecules at the site of detection. There are so many sensors out there actually, and so many people have already worked on developing very sensitive, ultra-sensitive biosensors. But the key bottleneck of all conventionally available biosensors is the diffusion-limited process. What I mean by diffusion-limited problem, every molecule has to diffuse to the site of detection by diffusion. Now, if the sample concentration is very low, the time it takes to diffuse from, let's say, from somewhere to the site of detection may take a few hours, if not days. So we want to solve this problem by using electrokinetic concentration. By using electrokinetic concentration, we can concentrate the molecules, all molecules, at the site of detection. And it allows us to detect this molecule of interest very quickly. And this is shown here. This is like a simple microfluidic chip. It has one reservoir here where we inject a sample into it. And then we use electrokinetic concentration to increase the local concentration. It's shown here. And you can see here, when you apply voltage across this microchannel, you can suddenly bring all the molecules, in this case is DNA molecules, in front of the membrane, in front of this conductive polymer membrane. And in this way, we can detect this molecule much faster and at very low concentration. Okay? And this graph shows you how fast we can increase the concentration. We start from around 10 nanomolar DNA concentration, and within less than 5 minutes, we can increase up to 10 micromole. There's around 1,000 times increase of concentration. So in this way, we can sort of increase the detection sensitivity of conventional biosensors and reduce the time of detection. Okay. Let me just show you a video clip. to demonstrate this electrokinetic concentration. You see here, this is a microchannel in the middle, and we apply voltage across this channel, and you will see a formation of fluorescence plug, which indicates the proteins. So we apply voltage. You can see here, it was black in the beginning, but after a while, you see this fluorescence plug formation inside this channel. So this is the concentrate proteins. So we can increase the concentration of proteins by several orders of magnitude. And in this way, we can detect this protein quickly. So our dream is actually to build a point-of-care diagnostics, a simple point-of-care diagnostics, simple but also sensitive. And we would like to deploy this point-of-care diagnostic tool in rural or in resource-poor settings. That's our sort of end goal. Now, I want to sort of give you a future vision, because you are all dreaming about making contribution to this field. And maybe not now, but maybe in 5 or 10 years from now, there will be a human on a chip in the market. Instead of doing all this testings in animals, maybe there's a chip where you can try all your drugs or new methods on a chip. So we call it human on a chip. You can envision building a chip where you can have some lung cells, heart cells, bones, kidney, and liver, all integrated on the chip, and you do all this testing. And it will save so much time and money, actually. And this, I think, is sort of my future vision, and I would like to give you this future vision and
So, which we switch gears back to more biology, but everything is actually connected, as you see. So I'm going to talk about basically how I really started getting interested in field of biology. And so my title is Take the Genome and the Environment, which basically sums everything that I do. Okay, so first of all, I'm going to ask a question. Anyone knows what is this?
You're pointing at the plant.
You see the little chicks here? Okay, what about now? Really? What is the prey bird, like falconers, here in UAE? Hobara, the Hobara, Habari, which is basically a flagship species here in UAE. So really, as an undergrad, I was really interested in this species. This is what I was doing in my weekend. I was basically following these birds in the desert of Morocco. And so this is a male Hobara. As you can see, it's a beautiful bird. This is a male displaying, touch females. I was also involved in hunting expeditions in the desert. Here, this is an example of what I was doing in my weekends. You can see here, I was actually taking this picture from my car, and you can see falconers. There is a falcon actually there, and that's a Hobara. So this is one of the favorite spots of chefs here in the UAE, also in the Gulf region. So they come always, like every winter, they come to Morocco to hunt these birds. So I was interested in taking parts of those, not because I was interested in hunting itself, but I was more interested in conserving the species. So that's how I got involved. And most of really what I was doing was working on those several species of birds. As I said, I'm a bird watcher, so I have my binoculars. Every Sunday morning, I go to the national parks or the desert and I observe those birds. Then I got really interested in this. I wanted to do something. So I joined this institution. It's called the International Foundation for Conservation and Development of Wildlife. This is the main entrance. And really the goal for this institution is to protect wildlife and also desert habitat and tradition. So this is just an overview of the institution. So I joined this institution as an internship just because I was interested in getting some training. This is the aerial view of the foundation. It's a captive breeding program. So we do a lot of research. You see the cages where we do captive breeding of the birds, of the falcons as well. I was based here in the genetics lab. So that's how I got interested in the field of genetics. So just to give you a very brief review of what we do in that place. So this is basically the process of captive breeding. So we have a male displaying, we do semen collection, then we inseminate females, we get eggs in the incubators, we have chicks, we hand feed them, and then the idea is to release them back in the field. And this is me, so I was spending, I would say, 2 days a week tracking those birds. Whenever we release them in the wild, we have them like with satellite tracking devices or radio tracking. So sometimes I spend 4 hours just to find one bird. And the idea is really to make sure that we are properly releasing those birds in the wild. And this is a conservation program. So the idea is to provide birds for the falconers, but also to do reintroduction to re-establish the population in the wild. So there are a lot of questions involved in this. At the time, I didn't have any expertise in genetics, but there are many questions here that we face. For example, when we collect semen from males, we inseminate different females. At the end of the day, whenever we have chicks, we don't know who's the father. And that was a big problem for this program. So the idea was to come up with a solution, who is the father? So that was really the haunting question for me. And then I had a discussion with somebody who's working in forensics lab, and he told me that's what you do for humans. So we had the idea, okay, can we actually use genetics to find who's the father? So for me, really, that's how it started. And so since then, I developed a lot of methods. There's just few examples. We can, for example, use genetics or DNA to find the sex of the birds. And this is a very important question. We can also monitor genetic diversity in captive breeding, because if you do captive breeding, you want to preserve genetic diversity. So that's what I was doing. And then I was interested also in this species, where we had a very interesting question. We had about 100 birds left in Morocco for this population, for this bird. But there are about 20,000 birds in Spain. So this is critically endangered population, but when we asked the Ministry of Conservation to put some conservation efforts, they told us, well, there are 20,000 species in the birds in Spain, why do we conserve those hundreds? We always get birds back from Spain. But we never really observed those birds crossing the straits. But we needed proof. So here again an example where we can use genetics. The problem is that we couldn't have any permit to catch the birds because they are highly endangered, so they didn't want to give us any permits. So I said, okay, can I use genetics to do this? The idea was to collect feces, because apparently you don't need a permit to collect feces in the wild. But it was a very challenging thing, so I spent 6 months in the lab. Basically, I developed a method to extract DNA from feces, and we were very successful, and we did genetic analysis, and we had proof that there are absolutely no movements between Morocco and Spain. That was a convincing argument for the Ministry of Conservation, and they decided to put some funding to conserve the species. So really, that's how it started. Around the year 2000, that was about the year where the human genome was published, and I started getting very excited by this field, a new field of genomics. It's the same idea as genetics, just at a bigger scale. So at the time, I was really hesitating. Do I keep working on conservation genetics and ecology, or do I switch? At the time, because of the human genome sequencing project, I was interested in doing something in humans, which basically was possible because there were a lot of opportunities to do this. So the question really at the time that I was interested in is basically to find genes that matter. And the idea is to find genes that we can associate to phenotypes. And I was more interested in disease. So how can we find genes that are associated with disease? So when I started my PhD, this question was probably the most interesting question at the time. But as you know, it's a long way to get from genes to a phenotype or to a disease. So in a sense, there is a black box between genes and phenotypes. So genes get expressed, then you have proteins and metabolites, then you have your phenotype. And then you have this big factor here, which is the environment. So I was interested in finding genes that matter, but trying to understand this in the context of the effect of the environment. So like I said, I was interested in humans. As you know, if you look at any two human beings, you will find differences between their genomes. But as we know, so there is a lot of variation when it comes to the environment. So different populations can be exposed to different types of viruses, they can live in different climates, they can eat different types of food, and also use different types of drugs. So really, the human phenotype, whether it's disease or not, it's a product of genes and the environment. So that was my interest. How to do that? I studied that in the context of urbanization, because urbanization is a major transition in lifestyle that the human population experienced in the last few decades. And also because we know that the incidence of complex diseases, you name it, cancer, asthma, diabetes are big problems. But this transition in lifestyle coincides with the increased incidence of those diseases. So really to understand those diseases, we need to understand the effect of genes and the environment. And that's how I will just give you an idea, for example, about how crazy those transitions are. This is Abu Dhabi as you know it, and this is Abu Dhabi 1953. So you can see that there is a major change in lifestyle in Abu Dhabi. Not just Abu Dhabi, but in many countries. This is Dubai. I tried to find a few pictures for Sharjah because we're not. But Dubai is another example where you can see this major change in lifestyle and urbanization. But because I'm Moroccan, I was interested in going back to Morocco and do some work there. This is my home city, Agadir, in south of Morocco, in 2014, and this is Agadir in the 1950s. So it's the same trend. So, and the Moroccan population is very interesting because it's a nice mix of different ethnicities. We have two major ethnicities, the Amazigh, or the Berber ethnicity, and the Arabs. And then you have some sub-Saharan genes also in the Moroccan population. But at the same time, we have three distinct lifestyles in Morocco. So you have very urbanized lifestyle, nomadic lifestyle, and in the mountains. So here you have a nice mix of genetic variation and environmental variation. And the idea was really to look at those variations. So I'm going to ask you to fasten your seat belts just for a minute. So I'm going to show you all the sites that I was working on. So this is Morocco, this is south of Morocco, this is the city of Agadir. As you can see, it's a very urbanized city. The density of the population is quite high. This is the site northern of Morocco, very close to the port. It's quite heavily polluted area because it's very close to the port. And we know that the incidence of diseases So this is still the city of Agadir. This is the other side, very close to the airport. It's a heavily populated area. This is all within the urbanized lifestyle. So I sampled the population in Agadir, and then to sample the second lifestyle, we're going to fly down about 100 kilometers south. So this is the small, that's actually the house of my parents. That was when I sampled the population, that was where I put all of my samples and tubes and everything. So this is the the rural lifestyle. This is the village actually where my grandparents lived. And you can see it's quite, it's a village. And it's in the middle of the valley. And then you have more villages. So at the time, there was actually no road here. It took us like 4 hours riding donkeys to get to the other village with all of our equipment. So you can see very isolated villages. And then we sampled the other villages on the other side of the mountains. And this is a very traditional lifestyle. They don't have any public services, no electricity, no running water. And lifestyle is all based on agriculture and grazing. So you can see some of the houses there. And then the last lifestyle is the nomadic lifestyle. So there are still a few hundred people living the nomadic lifestyle in Morocco. It was very hard to find them. This is basically, I have nothing to show you here. It's in the middle of nowhere. And we used to drive just trying to find 1/10 of good ones. But we were quite successful. We sampled about 20 nomadic families. So these are some pictures. This is from the village. This is from the nomadic trip. And you can see I have my lab, my equipment, and everything in the back of the car. This is one interesting family. There are actually 6 generations in this family still living. So the little girl has a grandma of her grandma still living with them. It's just fascinating. And I thought initially that it's going to take me 2 weeks to sample this. It took me actually a month and a half. Every time we sample a nomadic family, we had to stay, drink tea, and eat food. So I ended up actually spending my time on that project. Okay, so I'm not going to talk too much about the experiment that we did, but we always go back to the lab with the samples, and we use this kind of tools basically to assign genes and the gene expression and genotype of those patients. So these are some of the comparisons that we made. And then we can look at the genes. How many genes are, let's say, turned on or off depending on the environment? So that was really the idea. And then we can look at those plots and we see the number of genes. The take-home message here is that about a third of our genes are differentially expressed depending on where we live. That's really the take-home message. Things are very, very interesting when we look at females versus males. For example, between the city and this village, when we look at males, there are not that many genes showing differences. We look at, females, there are not that many differences. But when we look at the number of genes here, you can see that it's basically about the vast majority of those 7,000 genes here are differentially expressed. And this is purely because males in this village go to the city. So because males in the village go to the city, their genes are expressed like someone who actually lives in the city. So it's a fascinating example of how the environment can affect our genes. And then we can do things like this. We can look at build maps of genes. Red means the gene turned on, blue means turned off. And you can see that, for example, individuals living in this village have all of those genes turned off or turned on, basically with few exceptions. And then we can take this further. We can look at disease. What was interesting is that respiratory diseases or genes involved in respiratory diseases show a lot of differences between those lifestyles. And we can even push this further, and we can look more specifically, for example, between this village and the city. The number one group of genes that show differences are cardiovascular disease. And those are linked basically to differences in lifestyle between those populations. So the take-home message really from this is that the idea is that we want to understand the differences between, or the effects of genes versus the environment. And from this particular study, we estimated about 20% of our genes show differences because of the genetic ethnicity, and about 60% show differences because of the environment. And then we have this fraction where you have genes that show the effects of both the genes and the environment. And then, after this project, I was interested in looking at those questions in different contexts. This is something that I did in Africa, where we look at susceptibility to malaria. So the environment here being the infection. And so we basically discovered genes. We create maps that look like this, where we find genes basically that are very, very important in how those kids in Africa are either susceptible or resistant to malaria infection. So understanding those genes and creating this kind of maps have a lot of value, because we can, just by genotyping, looking at the genetic makeup of those kids, we can actually predict how they would respond to malaria. So if we have kids that we know are very susceptible to malaria, then we can do something about it. And more recently, we did this project in Canada where we sampled about 20,000 individuals, and we create these kind of maps where we try to understand really the effect of genes and the environment. This is the mitochondrial genome, which is an interesting genome in itself. The mitochondria is the powerhouse in our cells, and those differences we see between individuals, they actually predispose certain individuals to have higher metabolic rates than others. So right now, expanding this work here at NYUAD Abu Dhabi, and really the key word for my research is that it's very multidisciplinary. So the key word is probably genetics, but we use genetic techniques, we use bioinformatics, computer science, statistics, and also technology. So finally, I just would like to thank the really study participants who took part of those studies, and I'll be happy to take any questions.